This is the second video in the review of the nervous system. And in this one, we're going to go ahead and review how an action potential, once created at the trigger zone, is moved from there to the end of the axon. And then secondarily, how an action potential would get from the end of the axon across to another cell or to multiple cells, whether that other cell is a neuron or a muscle fiber or a gland or whatever it is that it happens to be. And so just in general, again, the action potential is created here at the trigger zone, and then it is conducted. That's the term that we use to indicate that it moves down the length of the axon. As you can see here, they're showing you a myelinated axon, and that means that the action potential will jump from the trigger zone to node 1, and then from there to node 2, node 3, node 4, and it will continue to jump like that until it gets all the way down to the synaptic knobs. Action potential conduction is also sometimes referred to as propagation. I tend to always use the term conduction, but you may see propagation as well. And so here what we're seeing is this is supposed to be the cell body of the neuron. And so here's the trigger zone, and then this is the axon. They're not doing a great job here, but what we have down at the end would be the synaptic knob. And then this is a second neuron that it will communicate with later. But what you can see is that we have indeed generated that action potential at the trigger zone. We know that because we look at the inside of the cell and it's positive when it's supposed to be negative. That means that the voltage-gated sodium channels have been opened up, and so we have sodium that has entered to the point where we make the inside of the cell all the way to positive. So roughly positive 30, positive 35. Now this part is a little tough to understand. It comes from physics and an understanding of electricity. But again, when we have these charges lined up this way, positive to negative, you know that unlike charges attract. Negative to positive attract, positive to negative attract. This allows the setup of the local flow of current. This is just like the flow of current that goes through an electric wire to power your computer or to turn on a light or to turn on your TV or whatever electronic device it is that you have. And so we have this flow of current that is going through this piece of membrane, which right now is still at the resting membrane potential. But as this flow of current goes through, it starts to stimulate the opening of those voltage-gated sodium channels. And so now you can see what's happening is we're starting to depolarize this adjacent piece of membrane. We're now looking at an axon which is unmyelinated. Well, if we get an action potential here at the trigger zone, we're actually going to open up enough voltage-gated sodium channels that we will get an action potential here in the adjacent area. And so sodium will flood in, and the inside here will also become positive. We move down to this piece of membrane, and you can see it's still at resting membrane potential. But once we have had enough sodium come in to create positive charges here, now we'll have the local flow of current from this new depolarized piece through to this adjacent unpolarized. It's at resting membrane. Well, that flow of current, again, it's electrical flow. It's going to stimulate voltage-gated sodium channels to open up. Sodium will begin to enter the axon at this point, and it will start to depolarize it as well. And if we get an action potential at the trigger zone, we are going to get an action potential here, and we will get an action potential here, and so it will continue up and depolarize all the way up to about plus 30, give or take a little bit. And so that process will continue. An action potential here will stimulate the creation of an action potential here. And the action potential here will stimulate the creation of the action potential here. And so we don't really move the action potential from the trigger zone down to the synaptic knob. We use the action potential at the trigger zone to create another action potential, another, and another, and another. And the truth is, we might create 1,000 action potentials or more to move from the trigger zone all the way to the end of the axon. And so that's what has to happen. The flow of current, represented by these small blue arrows, is moving through a piece of membrane which is at rest and stimulating the opening of the voltage-gated sodium channels so sodium can come in and depolarize that new piece of membrane. And so there you go. This is just a different depiction of the same thing. And so again, we've gotten our action potential here at the trigger zone. And we know that because we've got positive charge to the inside instead of the regular negative charge that we would have when the cell is at rest. And so now again, you can see that what's going to happen as a result of this, unlike charges attract. And so we're getting flow of current from positive to negative, but then from negative to positive, positive to negative, and all the way around. We have to have a complete circuit or we don't have the flow of current. That flow of current through here opens up those voltage-gated sodium channels, and in fact it will also open up the voltage-gated potassium channels, but again they just respond a little bit more slowly. So first we get depolarization and then we get repolarization, as we can see here. Now the trigger zone is repolarizing as this adjacent piece of axon is getting its action potential. Well, again, we're going to have a flow of current through here. And as it goes through this piece of membrane, which is at rest, it's going to stimulate opening of voltage-gated sodium channels first, and then secondarily, voltage-gated potassium channels. Oh, look, we've got a new action potential. This is action potential number 3. First was at the trigger zone. Second was just a little further down the axon. Third is even a little further down the axon. But now again, look, the piece right behind the part where we have the action potential has repolarized. We've opened up those voltage-gated potassium channels. Potassium is leaving and driving the inside of the membrane back to negative. Well, this process is going to continue. Even though they don't show you the rest of the axon, there's still some axon here that's at rest. We get that local flow of current. We stimulate opening of voltage-gated sodium channels first. Sodium comes in, depolarization. Then the voltage-gated potassium channels will open, potassium out, repolarization. And so that continues until we get all the way down here to those synaptic knobs or synaptic buttons. And it will continue into both branches, whether we're looking at collateral axons or whether we're looking at those telodendria. Here you can see again the same thing. Here's the trigger zone, labeled number 1. They call it axon hillock. I don't like that. It's not appropriate. Trigger zone is what we really need. That's where the action potential is first created. I hope you can see that this is telling us that we're at plus 30. We've depolarized. And so here the arrow is showing you that because of that we get this flow of current. And as it goes through this piece of membrane, it opens up those voltage-gated sodium channels, and you can see that we're starting to depolarize. It's at negative 60. And so I guess I didn't quite want to do that just yet, but that's all right. We'll move on. And so then this one will indeed reach positive 30 because we're going to open all the voltage-gated sodium channels. Once we do get to positive 30 here and we have positive charge inside, again, there'll be flow of current from this piece which is depolarized across to the piece which is at rest. And as the current moves through this piece of membrane, it stimulates opening of number 1, voltage-gated sodium channels. Sodium comes in. Number 2, voltage-gated potassium channels, potassium leaves, so that we get the complete action potential, the depolarization, the repolarization. And so that's what you have to remember. Now we move on and we look at the same process in an axon which is actually myelinated. And again, you know that the area of membrane where there is no myelin is referred to as a node. So here we can see trigger zone, node 1, node 2. They're not showing us any more nodes as we go down. We're just going to illustrate it here. What you have to understand is that when we look in the phospholipid bilayer here in the node, we're going to find voltage-gated sodium channels. We won't find those in the internode region. There's no point in having them. If we had a channel here and it opened up, the myelin would prevent any ions from moving through. We're also going to find voltage-gated potassium channels, and I hope by now you've also filled in the missing piece and you've said, well, we're also going to find we're going to find sodium-potassium pumps. Sodium-potassium pumps. And here you can see almost a complete diagram of that, but not quite. The green would be the voltage-gated sodium channels. The purple would be the voltage-gated potassium channels. And so you can see, unfortunately, what they've done is they've left out those sodium-potassium pumps. But now again, as we move down, the action potential at the trigger zone, look, plus 30, is going to set up this local flow of current which moves through the membrane at node number 1. That stimulates the opening of the voltage-gated sodium channels, and sodium comes in. Now, at the same time back here, voltage-gated potassium channels have opened up. Potassium is leaving so that we've repolarized that piece of membrane. As node 2 hits its peak of action potential, and actually even a little bit before that, we start to see that flow of current, which is going to move through node 3. Sodium is going to come in and will depolarize node 3. You can see that process has started because it's to -60. This should also help you to understand why the conduction of the action potential down a myelinated axon is so much faster. Again, it's like 200 miles per second that this action potential is moving, because rather than needing to get 1, 2, 3, 4, 5, 6, 7, 8 action potentials, we only need 2. And then again, we need 1 more, and then 1 more. And creating an action potential does take time. Well, if we can skip a piece of the axon before we create the next action potential, it's going to go faster. And so that's exactly what happens when we compare the conduction speed in an unmyelinated axon to the conduction speed in a myelinated axon. And so again, the local flow through the nodal membrane opens up the voltage-gated sodium channels and the voltage-gated potassium channels. It's just that, again, the potassium channels respond more slowly, don't open up quite as quickly. And then again, don't forget that those sodium-potassium pumps, once the channels are closed, have to pump the sodium out and the potassium back in. Now we need to talk about how do we get the action potential from one cell to the adjacent cell. And this is what we refer to as transmission. Well, the fastest way for us to do it is to use these gap junctions. Gap junctions are these protein channels that make holes in the membrane of cell number 1 and the membrane of cell number 2. And you can see that these gap junctions line up with one another. This means that cytoplasm of cell 1 is in direct contact with the cytoplasm of cell 2, and we get the flow of ions right through here. And so this is great. We have already talked about this in cardiac muscle tissue. And so here's that intercalated disc, which is the junction between 2 cardiomyocytes. Here we open it up and we see in this cardiomyocyte there are the gap junctions, and they will again line up with the gap junctions in the cardiomyocyte that sits on top of this. And so as we get an action potential in this cardiomyocyte, it is directly transmitted to the cardiomyocyte that's been taken away. And so not only do we find these gap junctions in the heart, but we've found more recently, the past decade or so, that we have gap junctions connecting some neurons in our brain. Not a lot, but a few. We also have them between glial cells and neurons in our brain. And then we find them in the liver, we find them in the pancreas, and we also find them connecting different smooth muscle cells within the GI tract. So that, again, that action potential is transmitted incredibly fast. And this is really what's referred to as electrotonic transmission. Okay, this shows you a better depiction of how many of those gap junctions exist and how densely packed in they are. Look at this. Here's a nice electron micrograph of this. And so you can see this is actually like creating a large hole in cell number 1, and it's going to be paired up with a large hole in cell number 2. Here's cell number 1, here's cell number 2. And so again, because we have this large flow, we get the flow of ion right through, and the action potential is created incredibly fast. Just really, really fast. More commonly, what we see happening is we see what's referred to as chemical transmission. Chemical transmission requires the release of neurotransmitter. And before we look at that, we're going to try and get an understanding of what's happening. You have to remember that in order to depolarize or repolarize a membrane, we have to change membrane permeability. And we do that using channels. Here's a channel. And this is just a schematic representation. Well, this channel is going to be opened up by the neurotransmitter acetylcholine. And as you can see here, it requires 2-acetylcholine to open this guy up. And so this channel is referred to as a ligand-gated ion channel. It's ligand-gated because it requires a chemical signal to open it up, the ligand. And in this case, ACH is the ligand. It's an ion channel because once we open it, sodium comes in, but potassium will leave. It's not specific to one ion or the other. Notice, please, that more sodium is coming in than potassium is leaving. This is just a different depiction of exactly the same ligand-gated ion channel. It opens up in response to the binding of 2 acetylcholines. Those are the neurotransmitters. And as soon as that happens, sodium is going to move in and potassium is going to move out. And again, more sodium goes in. It's important that you understand that. It's also important that you understand that the neurotransmitter does not move through the channel. Neurotransmitter stays out here in the interstitial fluid, and it will be dealt with later. We get rid of it later. Here's a slightly different ligand-gated ion channel. Unfortunately, they're not naming the ligand for us, but you can see that it's going to open up a calcium channel, and calcium will move from the interstitial fluid through into the inside of the cell, because that's the way that calcium will go. Higher concentration for calcium outside the cell than inside the cell. Okay, they've done a better job of really representing what a protein looks like. You can see both the secondary and tertiary structure of this ligand-gated ion channel, which is protein. That's what it's made from. Here's one that's slightly different. And so now it responds to the neurotransmitter GABA, which is actually an inhibitory neurotransmitter. As the GABA binds to its receptor, then the channel opens up and chloride will move through, bringing in negative charge with it. And so this is one way that we can actually hyperpolarize this membrane. It's making it more negative inside than it was before. And so you can see that in these cases, the ligand-gated ion channel is not only the ion channel, but it also is the receptor for the neurotransmitter. And again, I'm going to reiterate, please notice that the neurotransmitter does not go through the channel, does not go through the channel. Okay, and then here's a different depiction of this GABA channel. And so GABA is going to bind, it will open up, and once it does open, then the chloride can move through, and it brings in more negative charge, and it allows the membrane to be hyperpolarized. Now, the truth is that not all ligand-gated ion channels are receptor and channel in one. Here's a different example. Acetylcholine can work with different receptors. And so here's a protein that has the receptor for acetylcholine. It binds, and what happens is we have to activate the G protein. I hope you remember something about G protein from when we did the nervous system and talked about it in human anatomy and physiology. The G protein then is going to open up a separate protein, which in this case happens to be a potassium channel. Potassium is going to leave the cell. It's going to go from the cytoplasm out to the interstitial fluid, it's going down its concentration gradient and taking positive charge away, which will also hyperpolarize this membrane. So here are two ways that we can cause hyperpolarization. Let chloride come in, or let potassium leave the cell. So either way, the same thing can happen here. And so now we've reviewed how you can go ahead and change the membrane permeability by opening up ion channels. Here's a different one that requires G protein for it to work. And so the receptor for norepinephrine is here. Again, this is a protein, but it's not also a channel. The channel is over here. It's a separate one. And so the G protein is going to work through the second messenger camp, and that's going to open up this ion channel. And again, they're not telling us specifically what kind of ion channel is it. Is it chloride, potassium, sodium? We don't know. It really depends on where it is that it's located. All right, and now we're going to go ahead and we're going to look at the more common type of transmission, chemical transmission, and here indeed is a chemical synapse. Well, the chemical synapse is the neurotransmitter, or the ligand. In here you can see there's the neurotransmitter in this case. This is a blow up of what's happening right there. And so we can see two neurons, and neuron number one is right here. All we're seeing is the telodendria and the synaptic knob. We don't even know if the axon that is connected to this is myelinated or unmyelinated. It doesn't matter. Either way, the action potential is traveling down the telodendria to get to the synaptic knob. That's what's going on here. And so you need to be able to identify the different pieces that we have here as we look at this. And so these guys are voltage-gated calcium channels. That's what they are, voltage-gated calcium channels, as opposed to the ligand-gated calcium channel that we just looked at. Voltage-gated calcium channels over here as well. Here are the synaptic vesicles. They're little bubbles. They're just like little tiny soap bubbles. That's exactly what they're like, because they're made up of phospholipid, and so they're a little bit soapy and oily as well. And here you can see we've opened up a few so that you can see that neurotransmitter inside there. And the neurotransmitter is going to move across and bind with the receptor. And so again, what we have here is we have ligand-gated ion channels, which are receptor for the neurotransmitter and channel all in one protein. Ligand-gated ion channels, and you can see that there are a lot of them, and they are found in the membrane of neuron number 2, which is on the other side of the synapse. So here's the chemical synapse. The synaptic knob, the synaptic cleft that we have here in the middle, and then the postsynaptic cell or membrane that we have there. Okay? Well, again, the green dots represent the individual molecules of neurotransmitter, whether it's acetylcholine or GABA or aspartic acid or whatever it is that it happens to be. The calcium coming in here is going to make these synaptic vesicles migrate or move down towards the end of the knob, and then they're going to also dock or merge with the membrane. Again, you got to remember, this is phospholipid bilayer, this is phospholipid bilayer, and those things can merge. And when they do, the vesicle opens up and releases neurotransmitter via exocytosis. And so the neurotransmitter is moving into the synaptic cleft right here via exocytosis, and then it will diffuse across, and it will bind with the receptor, and it will open up the ligand-gated ion channel. In fact, I know that this one is not acetylcholine because it takes 2 acetylcholine to open up the ligand-gated ion channel here. But once it's open, sodium is going to move from the interstitial fluid here in the synaptic cleft through the open ligand-gated ion channel and into the inside of the postsynaptic neuron. This is phospholipid bilayer right here. This is the postsynaptic neuron. That's what this one is. And so as the sodium comes in, it begins to depolarize it. At the same time, potassium is leaving. But look, the sodium arrow is much bigger, indicating that more sodium comes in, and so we don't have a one-for-one change of positively charged ion. We've got more positive charge coming in. We're going to depolarize the postsynaptic neuron. That's what we're going to do right here. Okay? And so that's what's happening as we do this. The result of this is we create what is called an EPSP. And so I don't know that this is a great picture, but that's what's going to happen. Here's our presynaptic neuron, there's the synaptic cleft, and here is our postsynaptic neuron. And it could be the dendrite, but it doesn't in fact have to be, as we'll see later on. Well, the neurotransmitter moving across is going to open up the ligand-gated ion channels. Sodium comes in and we depolarize. Oh, look at that. And we create an EPSP. This is an excitatory postsynaptic potential. So it's like an action potential in that we're depolarizing, but it's different because, look, we're only depolarizing a little bit. We didn't reach threshold, so we don't get an action potential. If this EPSP is only about 10 millivolts, we get to negative 60, maybe to negative 62, I don't know, something around there. And then sodium stops coming in. The sodium-potassium pumps pump that sodium back out. They pump in the potassium, and we quickly return back to the resting membrane potential. Now, on the other hand, if we had opened up those GABA channels, don't forget what's going to go through is chloride. And chloride is going to bring negative charge with it, and that chloride is going to create an IPSP. Well, look at that. Inhibitory postsynaptic potential is actually hyperpolarizing. What have we gone to? We've gone to approximately negative 80 millivolts. And so, again, we're constantly creating EPSPs and IPSPs. And whether or not we get an action potential in the postsynaptic neuron really depends on how many EPSPs did we get, how many IPSPs did we get. And so here you can see what happens. Look, if we get 2 EPSPs, but the second one occurs after we return to rest, we still don't get any action potential. Now, in this case, we've got 2 coming quickly, one right after the other. And so the second one begins before we returned to negative 70, the resting membrane potential. They add together, we hit threshold, oh, we get a full-blown action potential. There it is. Depolarization because those voltage-gated sodium channels are open. Sodium is rushing in. We reach the peak. The inactivation gate of the voltage-gated sodium channel shuts. The voltage-gated potassium channel is now open. Potassium leaves, we repolarize, and we even get our undershoot here. And we go a little bit past negative 70, and now the voltage-gated potassium channels are shut. Sodium-potassium pumps are moving stuff back where they belong. We get back to our negative 70 resting membrane potential. Here you can see a case where 2 EPSPs arise from 2 different presynaptic neurons. You're going to have lots and lots of presynaptic neurons here. And so we get an EPSP from neuron 1 and we get an EPSP from neuron 2. They arrive at the same time. They add up. We hit threshold. We get an action potential. Now, in this case, we get an EPSP from neuron number 1, but we get an EPSP from, we get an IPSP from neuron number 3. And now the EPSP and the IPSP add up, but negatively. The IPSP is going to subtract from the EPSP, so it doesn't get anywhere near threshold, and we get no action potential. And so inhibition. And that's exactly what happens to motor neurons that go to your triceps when you're doing a biceps curl. You don't want your triceps to contract when you do a biceps curl. And so back there in the spinal cord or in the brain, we're going to get IPSPs to the motor neuron going to the triceps to help keep that little bastard quiet. Okay? Here we can see a specialized chemical synapse. And if you look at it, you might recognize it. Oh my gosh, look. Here is the alpha motor neuron to a muscle fiber. And so this thing is a neuromuscular junction. That's what it is. Neuromuscular junction. It's just a specialized chemical synapse. It has the same 3 parts that we had before. Here's the synaptic knob. The synaptic knob is nice and big because we've got literally thousands of synaptic vesicles in there. The other thing that I forgot to mention before is that, again, a neurotransmitter is released via exocytosis, which is an energetic process. So we also have to have lots of mitochondria in here. And the synaptic vesicles, the mitochondria, they take up space. They make the synaptic knob bigger than the telodendria. The second part is the synaptic cleft. Here's the cleft, which again is full of interstitial fluid. That's what we have right there. Okay, and then finally the last part in this case is specialized muscle membrane, sarcolemma, and so it's the motor end plate. In the nervous system, it would be the postsynaptic neuron. But that's what we have. Again, this is phospholipid bilayer, and in here we can see the ligand-gated ion channels. And so super, there you go. All right? And so here's the action potential arriving. Here are the voltage-gated calcium channels that are opened up by the action potential arriving. Calcium will diffuse in because there's a higher concentration of calcium out here in the interstitial fluid. That's a terrible A, isn't it? It looks kind of like a Q or a 9. I can't erase it, I'm sorry. But at any rate, the calcium is going to go down through because there's way more calcium out here in the interstitial fluid than there is inside the cell. And again, that calcium causes migration and docking, which is a very complex process. So we're not going to get into how that happens. I just want you to know vesicles move down and they merge, they open up and they release neurotransmitter via exocytosis. In this case, the neurotransmitter, the little red dots, is actually acetylcholine. That's what it is, acetylcholine. It takes two of them to open up a ligand-gated ion channel. And so you see that the acetylcholine is released via exocytosis. It diffuses across. Two of them will bind with a ligand-gated ion channel, and the ligand-gated ion channel will open up. And as a result of that, sodium is going to diffuse through, potassium is going to leave, but again, we've got more sodium coming in. More sodium coming in because we have both the concentration gradient, high sodium concentration outside, low sodium in here, but we also have negative charge, which attracts the positively charged sodium. Sodium is going down its electrochemical gradient. Potassium is only going down its chemical gradient. It's got a positive charge as well, and so the positive charge here is trying to keep it inside. At any rate, as the sodium comes in, it creates a special EPSP. Since it's in the muscle, we call it an end plate potential. But this is just simply a specialized EP. See how well I can do this? SP. And so an end plate potential is just simply a specialized excitatory postsynaptic potential, post on the other side of the synapse. That's what it is. Well, then the EPP is going to move along the sarcolemma, and it's going to reach these voltage-gated sodium channels, and it's going to stimulate them to open up. Okay, they're in the membrane of the sarcolemma adjacent to the motor end plate. That's where they are. And of course, what's missing? Pause the video and tell me what's missing. And so what you should have said is that we're missing our voltage-gated potassium channels. Those are going to be here as well. And we're also missing the sodium-potassium pumps. But once we get the action potential here, it's going to go zooming out across the entire sarcolemma in all directions. It's going to go right, left. It's going to come towards us. It's going to go into the computer screen. And it's very quickly going to travel throughout the entire sarcolemma. Okay, and so we get that action potential that goes in all directions. Here's just our reminder of what that voltage-gated sodium channel looks like. Here it is when the sarcolemma is at rest. inactivation gate is open, activation gate is closed. Here it is when it's been stimulated by the end plate potential to open up. Activation gate opens up. The inactivation gate shuts, but it does it very slowly. And there's plenty of time for sodium to move through and cause the action potential before the inactivation gate shuts. And then lastly, here we just look at some of the different types of chemical synapses we can have. Here's what we might consider the most common. This is an axodendritic, axon to dendrite, axodendritic. Here is one which is a little bit less common, but still common, axosomatic. Here's the axosomatic one. And here's one that's a slightly rarer. We have axoaxonic. And this is, in fact, where we can get some IPSPs being created to try and prevent any action potential occurring here in the trigger zone. And here's a nice photomicrograph of the axon synapsing with a postsynaptic neuron. And then the truth is that even rarer, we've finally found that we even have a few cases where we have a dendrodendritic synapse. And so that can happen as well. That brings us to the end of video number 2. In video number 3, we're going to start to look a little bit at how the brain can start to control some movement. We'll look at how information gets from your cortex down to your spinal cord, and then from the spinal cord out to the muscles.